everyone, welcome to our topic 7 assignments this week. So some of you still haven't taken the midterm exam, so there's still a couple of dates that I'm proctoring the exam. If you haven't signed up for either of those dates and you have not taken the midterm exam with me yet, then you need to sign up via ProctorU and complete the exam through them. And that will be, I think the deadline for the exam is Wednesday midnight, so you got to do it by Wednesday midnight, otherwise you'll lose the opportunity to take the midterm exam, which is worth quite a bit of your grade. So do complete that ASAP. And then for those of you that are taking it with me, I will see you either Tuesday or Wednesday. So this week we're going to take a closer look at genetics. So last week we looked at mitosis, which is where we take our cells and we make exact copies of them through that cell division process. So this week is a little different because instead of creating daughter cells that are identical to the parent cells, we're going to learn about meiosis, where the daughter cells are genetically unique from the parent cell. And so we'll learn about how that occurs, how we go from a diploid parent cell to 4 haploid daughter cells that each contain a unique combination of the parent cell's DNA. And we'll apply that to inheritance patterns. So, thinking about your own traits, half of your DNA came from your mom and half came from your dad. So that means you have a mixture of their DNA. And so you can see certain traits that you inherited from your mom and certain traits that you inherited from your dad. So we'll learn why that occurs, why, maybe your eye color is more similar to mom's, but your hair color is more similar to your dad. So we'll learn about those inheritance patterns. So this week, just be aware that we will do half of topic 7 this week and the rest on next week. So when you are looking up your student roll, just be aware that you'll have the same student roll this week as next week. So if you were assigned student number 1 this week, you'll be student number 1 next week as well. Okay, so let's take a closer look at our due date calendar. So we've pretty much caught up, right? So we're continuing everything through. We do have our the topic 7 assignments starting today, and the topic 6 quiz due by Wednesday midnight, as well as that's when the midterm exam closes. Topic 7 assignments. This week you have 4 of them, and those 4 will be due by Sunday midnight. And then the enzyme concentration lab report that you collected the data for last week, that's you should be continuing working on that lab report and preparing to turn it in the day after the topic 7 assignments are due. So topic 7 S1 through S4 are due Sunday midnight. Your lab report, your second lab report, is due by Monday midnight. Okay, and then in addition to all of that, we should be spending some additional time on our integration project. So I'll talk a little bit about that as well in this video. So as always, it's a good idea to start with Dr. Blackwell's summary of topic 6, as well as the beginning of topic 7. So he has 2 videos. The first one applies to this week. The second one applies to next week. So you can come back and visit that later. We have our Adigia textbook assignments, and those, let me just take a quick look at the Adigia textbook assignment. So it's meiosis and inheritance patterns. So you have 2, 4, 6, 7, 8, 9 questions that go along with that. I won't be checking your Adigia quiz grades until the end of week 2. Okay, so the end of the following next week. just because you might, some of it applies to this week, some of it applies to next week. Okay, so just be aware of that. Your grade won't be updated. Let me go back to topic 7. Okay, and then of course your student roles will be different this week than last week, so do make sure you check and see which one you were assigned. All right, so let's take a look. I have my workbook in front of me because I don't have the digital copy for the remainder of the term. So I'll be using that as kind of my guide of what we're doing this week in each one of these Moodle assignments. So the first assignment, before you even get into it, always a good idea to take the reading and try to answer the preparation objectives on page 225 and 226 from your Digio textbook. That way you can kind of get us some background information before you get into some of these difficult topics. So you're going to start out by using the readings from the Digio textbook as well as this little mini lecture. So the mini lecture kind of goes through the process of meiosis, okay? And because a lot of us are more visual learners, I highly suggest that you also maybe refer to the Amoeba Sisters or the Crash Course Biology YouTube videos on meiosis. Okay, so you just go to YouTube and click meiosis, Amoeba Sisters or meiosis Crash Course, and you'll get access to those videos, and they're pretty good summaries and visuals of the process of meiosis. Once you have kind of a grasp on it, and then you're going to work your way through the activity here. So part of the activity, you're going to be taking a look at chromosome 7 and chromosome 15. So I highly suggest that when you're looking at chromosome 7 and 15, particularly on page 229, you cut them out. Make sure you color them, okay? So it doesn't have to be like, I think in the book it tells you to color blue for the fathers and pink for the mothers, but if you don't have those colors, just use anything, two different colors, and that way it'll help you keep track of the chromosomes and the particular genes that are found on the chromosomes as you complete the activity. Let's see, anything else? Yep, so you'll be looking at the results of meiosis with those chromosomes travel through the different stages of meiosis and then what they ended up in the different gametes at the end. And then you have those summary questions that kind of summarize everything. So once you've completed the simulated little meiosis chromosome activity and the questions that are associated with it, go ahead and post your responses by Sunday midnight. So page 237 you can ignore, so that is for in-person where they're going to prepare for extracting their DNA. We won't do that. But you're going to get into page 239, and 239 is going to get into inheritance patterns. So now that we know why our chromosomes are, how they isolate during meiosis and end up in the gametes, we're going to take that information and we're going to apply it to inheritance. So if, the sperm of one from the father parent has a colored gene that says black, and the egg of the other parent has a color gene that says brown. When sperm and egg unite, and we have two genes now, one says black, one says brown, how is it going to be expressed? Okay, so you'll go through this with this experiment with dog breeding, and we have the black dogs and the brown dogs, I believe. Yeah, I think so. And see, how that occurs. So again, the Adigio reading will help you kind of understand the background, and then you'll apply what you understood from the reading to this activity with using dog genetics as the basis. So you'll play around with that idea and draw, what goes on inside each of the cells and so forth, and see what the possible outcomes are. As you go through, just be very, very careful with naming things. Okay, so if we're talking about one single gene, we always use just a single letter from the alphabet, and it has to be the same for both the homozygous and the heterozygous, or the homozygous dominant and the recessive allele. So for example, it talks about using the letter B. Okay, so uppercase B indicates dominant allele, lowercase b indicates recessive. Sometimes I see students, they use different letters from the alphabet. Okay, so they might use B and then W or something like that. It gets very confusing when you're using different letters of the alphabet. So always indicate the uppercase letter for the dominant allele and the lowercase letter for the recessive allele for a single gene. If we have two different genes that we're talking about, those would have different letters of the alphabet. But when we're talking about a single gene, we want to use a single letter of the alphabet. Okay, so just be aware of that. If you get into the habit, it makes a lot more sense later on when we get into dihybrid crosses and things like that. Okay, so you're just going to be working your way through the different examples. These can be a little complicated at first. So again, Crash Course videos and Amoeba Sisters videos, they both have good videos on patterns of inheritance. That's basically what you're going to look for. Patterns of inheritance, or Mendelian genetics is the other key term. And they'll walk you through how to set up a Punnett square and things like that. So once you've completed the assignment, go ahead and post it. All right, and then the third activity this week is, again, we're not actually extracting our own DNA for this third lab report like the in-person classes do. But we still have to understand the whole process. So the in-person classes this week, they are going to actually extract their DNA. So they're going to be taking a toothpick and scraping the inside of their cheek cells and placing it, actually I don't think they use a toothpick. They use Gatorade, I think, and swish it around in their mouth and then spit it into a cup. And then they go through a process of DNA extraction to remove the DNA from the cells inside the Gatorade solution in the cup. And so you'll learn about how that occurs with this DNA extraction information. Okay, so there's a little video to learn how it occurs. So again, you got to do that on your own. Also this week, when the students finally extract their DNA, they've basically caused the cells, their cheek cells, to burst open, and they've been able to isolate the DNA strands. They're going to take that, and later the instructor actually amplifies the DNA that we're looking for. In this case, we're looking for that bitter taste gene within all of our DNA. And so they amplify that particular bitter taste gene through a process known as polymerase chain reaction. So you'll learn about this process in this virtual lab here. Okay, all about that. So you can watch the videos and then kind of go through the different parts. Okay, so even though, again, we're not going to actually be completing all of the different parts of this activity in person, we still really need to now understand how it occurs. So you'll be working through the pages from your workbook, so page 252 all the way to the next page, 253, using what you learned from the virtual labs to answer the questions. Okay. Go ahead and post your results there. And then finally, we have our human genetics. So in the previous activity, T7S2, we looked at dog genetics and things like that. So this one, we're going to focus on human genetics. So we'll look at things like polygenic inheritance. So basically traits that have more than one gene involved. We'll also look at basically the genetics between males and females. So the XX versus XY. So what's so important about that? In particular, we'll look at sex-linked diseases. So mutations that occur on a gene on the X chromosome tends to end up having more issues in males than females, because males only have one X chromosome. So we'll learn about those sex-linked traits and things like that, and how to undergo the Punnett squares. Excuse me. Okay, and then on page 261, you'll see some examples of karyotypes. So a karyotype is just a display of the chromosomes from an individual. So you'll see, if you counted up all of the chromosomes in the karyotype, it'll add up to 46 for a normal human. And that means that we have 23 pairs of chromosomes. So you can see that we have a pair of chromosome number 1, we have a pair of chromosome number 2, and so forth. So you'll see the difference between a normal male and a normal female karyotype, and then you'll also observe some abnormal karyotypes. So you'll see basically what happens when we have an extra chromosome. If it happens to fall on an autosome, chromosome 1 through 22, it will have much different effects on the body, more extreme effects, than say if it was if the abnormality occurred with the sex chromosomes. Okay, sorry about that. So again, with the karyotypes, it's very interesting to see how it's even formed. How does a person end up with an extra chromosome, or perhaps missing a chromosome? So you'll learn some of that information this week in that assignment. Okay. Let's see. So that was human genetics. Now it also says spend some time on the integration project. So each one of you signed up for an integration project topic way back in topic 4. Okay, so if you haven't had a chance to do that, I assigned you one already. So you can check and see which one you were assigned. If you decided, this week that you didn't like the one that you chose, you can still change it, but you have to email me and let me know which one you've chosen. And remember, it cannot be the same as anybody else. That's, we can't have duplicates. Okay. So because we're learning about genetics this week, it's a good idea to do some research on your particular genetic disease integration project topic, just to see, which chromosome it occurs on, and what's the inheritance pattern. Is it a dominant or recessive allele that contributes to the disease, and things like that. So you can do some of that background this week, which will, even though you're not bringing together your project until week 10 is when it's due. You might want to start working on some of that background this week. Okay. Now you'll also notice that this week there's no quiz after, because we go into next week. Okay, so the only quiz that you have to prepare for is the one that's due this Wednesday on topic 6. There won't be one next week because we'll do all of topic 7 quiz the following week. Okay. And then going back, you should also be working on your topic 5 S6 enzyme lab report. So I do have a video. Okay, so I highly suggest that you watch this video. So I go through each part of the second lab report, what you should include and everything like that. And then once you've written your lab report and you're ready to submit, don't forget to grade your lab report using the rubric. So I'm in instructor mode, so I can't show you, but it'll look a little different in your view. Well, you'll have the rubric right below it, below where you submit it. So be sure to check that before you actually submit, so you don't leave anything out. Okay. Let's see, anything else? If you have anything that you're concerned about or confused about, feel free to email me. We can always set up a Zoom meeting and go through it together, or we can just email back and forth. Okay. Otherwise, good luck this week. And for those of you that haven't taken your midterm exam, I'll see you either Tuesday or Wednesday. Take care.